traveled all around the cell to understand cellular respiration and photosynthesis. The four main stages of cellular respiration are glycolysis, pyruvate oxidation, Krebs cycle, and the electron transport chain. Glycolysis happens in the cytoplasm of the cell. Pyruvate oxidation happens as the products of glycolysis are entering the mitochondria. Krebs cycle happens in the mitochondria, and the electron transport chain happens in the inner membrane of the mitochondria. Glycolysis starts with one molecule of glucose. Then, an ATP gives up an inorganic phosphate to create glucose 6-phosphate or G6P, with the help of an enzyme that is rearranged to fructose 6-phosphate or F6P. Then, another inorganic phosphate is given by ATP to create fructose 1,6-biphosphate or F1,6BP. The F1,6BP then splits into the hydroxyacetone phosphate or DHAP and glyceraldehyde 3-phosphate or G3P. With the help of an enzyme, the DHAP is transformed into G3P and the process continues with two G3Ps. Hydrogen atoms reduce NAD+ to create NADH and 1,3-biphosphoglycerates or BPG. One ADP molecule comes to take an inorganic phosphate and make an ATP and 3-phosphoglycerate or 3PG. An enzyme helps to rearrange 3PG into 2PG. Water is extracted to create phosphophenol pyruvate or PEP. And finally, an ADP comes and picks up an inorganic phosphate to create ATP and the final product, pyruvate. Let's do the math. 2 ATP were initially invested. 2 ATP plus 2 ATP plus 2 NADH were gained. The 2 invested ATP will cross out with the 2 gained ATP, and in total, in glycolysis, we produce 2 ATP and 2 NADH. The next step in cellular respiration is pyruvate oxidation. In pyruvate oxidation, pyruvate produced in glycolysis travels from the cytoplasm into the mitochondria. As the pyruvate travels across the membrane of the mitochondria, 3 main steps happen. Firstly, CO2 gets extracted. Then, an NAD+ is reduced, and a proton is released. And finally, coenzyme A attaches to the molecule to produce acetyl-CoA. Let's do the math. In pyruvate oxidation, we start with 2 pyruvate and end up with 2 acetyl-CoA and 2 NADH. Therefore, in total, the products of pyruvate oxidation are 2 acetyl-CoA and 2 NADH. The third step of cellular respiration is Krebs cycle, which starts with one molecule of oxaloacetate. The oxaloacetate goes through a condensation reaction with the acetyl part of acetyl-CoA to make citrate. The citrate is then rearranged into isocitrate. A CO2 is extracted and an NAD+ is reduced to create alpha-ketoglutarate. The same steps repeat, and a coenzyme A is attached to create succinyl-CoA. The CoA is removed, and a GDP gets phosphorylated to GTP, which is used to create an ATP. The product is succinate. An FAD gets reduced to produce fumarate. A water is added to create malate, and finally, an NAD+ gets reduced to go back to the initial oxaloacetate. To summarize, there are 8 different molecules involved in Krebs cycle: oxaloacetate, citrate, which gets rearranged to isocitrate, alpha-ketoglutarate, succinyl-CoA, succinate, fumarate, and malate. Let's do the math. In Krebs cycle, we invest 2 acetyl-CoA and gain 3 NADH, 1 FADH, and 1 ATP. Since Krebs cycle happens twice, we actually end up with 6 NADH, 2 FADH, and 2 ATP. The final step of cellular respiration is the electron transfer chain. The main 3 proteins involved are NADH, deoxyhydrogenase, cytochrome BC1 complex, and cytochrome oxidase complex. The most important one, of course, is ATP synthase. The electron carriers are ubiquinone and cytochrome C. When NADH is oxidized, the electrons enter through the first protein. As the electrons pass through each main protein, a proton gets pumped into the intermembrane space. The electrons leave the chain to join a reaction involving oxygen and protons to make water. The concentration of protons in the intermembrane space is high, therefore the protons get pumped back. Since they're going with their concentration gradient, energy gets released and ATP gets made. What happens to FADH? FADH passes its electrons to ubiquinone instead of the NADH dehydrogenase. Only 2 protons get pumped out and only 2 ATP are made. In the electron transfer chain, NADH produces 3 ATPs, while FADH produces 2. In glycolysis, pyruvate oxidation, and Krebs cycle, 4 ATP, 8 NADH, and 4 FADH get made. There are 4 initial ATPs, 24 from NADH, and 8 from FADH, and in cellular respiration, the total number of ATPs produced is 36.
I'm sexy and I know it.
Introducing photosynthesis.
The light reactions in photosynthesis are very similar to the electron transfer chain in cellular respiration. The light reactions use 4 main proteins and ATP synthase, and they happen on the membrane of the thylakoid. The 4 main proteins are photosystem 2, P680, B6F complex, photosystem 1, P700, and lastly, NADP reductase. There are also electron carriers like plastoquinone, plastocyanin, and ferredoxin. On top of photosystem 1 and 2, there are things called antenna complexes, which are composed of pigments. When hit by light, the electrons in the pigments get excited and jump up. They get caught by a primary electron acceptor and get passed on to plastoquinone. At the same time, the Z enzyme breaks down water to create oxygen, protons, and 2 electrons, which replace the electrons lost in the photosystem 1. As the electrons pass through the B6F complex, 4 hydrogens get pumped in. The same process happens in the second complex. The initial electrons replace the ones lost here. The electrons are carried out by NADP reductase and an NADPH is produced. The protons that are now inside the thylakoid get pumped out by ATP synthase and the ATP is produced. The NADPH and ATPs then continue on to Calvin cycle. The third reaction, or Calvin cycle, is very similar to the Krebs cycle. It starts with 3 ribose 1,5-biphosphate, or RUBP. The 3 RUBP join with CO2 and H2O, and with help of Rubisco, 6 3-phosphoglycerates, or PGAs, are produced. Using energy from ATP, the 6 PGAs are rearranged to 1,3-biphosphoglycerate, or BPGs. To get the 6 wanted glyceraldehyde 3-phosphates, or G3Ps, 6 NADPHs are oxidized, and 6 inorganic phosphates are extracted. 1 G3P exits and can be used to make glucose, starch, or sucrose. The other 5 continue to get reassembled to RUBP using 2 waters and 3 ATPs. In conclusion, Calvin cycle can be split into 3 phases: carbon fixation, reduction reactions, and finally, regeneration of RUBP. Using sunlight, the light reactions produce ATP and NADPH. These are used to drive the Calvin cycle. In Calvin cycle, we invest 9 ATP and 6 NADPH to make 1 G3P. Since 2 G3Ps are needed to make glucose, we actually invest 12 NADPH and 18 ATP.